Hello everyone, I am back today actually for quickly revising all the concepts we have studied in the cell, the unit of life. Okay, so let us begin or quickly recapitulate everything which we have discussed till today. Okay, so first thing is organisms can be classified into two types basically on the basis of number of cells they have, unicellular and multicellular. Okay, both of them follow independent existence and all the essential activities are carried out inside the cell. Okay. Next thing is division of labor. It is found only in the multicellular organisms. It is not found in unicellular organisms. Division of labor basically is that different types of cells are assigned with different kinds of functions. They perform those functions specifically. Not a single cell perform all the types of functions. Okay. Next comes what are the things which are required to be a cell. It is basically three things: cytoplasm, cell membrane and nucleus. If all the three things are present, then the cell is living. Okay. Now what about RBC and sieve tubes? They are basically exceptional cases in this thing because both of them lack nucleus. Okay. Now next thing is, what is cell? Cell is basically the structural and functional unit of life. Okay. Now let us discuss about the next. Now Robert Hooke was the first scientist who saw a dead cell wall. Okay. He basically took sample pieces from Spanish oak tree, and its scientific name is Quercus suber, and he observed the beehive like structures and named it cella, about which he wrote it in his book Micrographia. Okay. Then next thing comes is, who was the first scientist to see a living cell? It first time Anton von Leeuwenhoek saw a living cell. Okay. In the sample of rainwater, he saw microorganisms moving and named it animalcule. Okay. Next thing comes is the cell theory, or before that Robert Brown saw the nucleus. Now in 1838, scientist Matthias Schleiden, who was a German botanist, observed that all plants were made up of cells and its products. In 1839, British zoologist named Theodore Schwann observed that all plants as well as animals were made up of cells and its products. So they gave the first point in the cell theory, which is all living organisms are made up of cells and its products. Now in 1855, Rudolf Virchow came and said omnis cellula e cellula, which meant all cells arise from pre-existing cells. So it was the second point of the cell theory, and it was completed by the Rudolf Virchow. So who was the scientist to complete the cell theory, modify the cell theory and give the cell lineage theory? It was Rudolf Virchow only. Now next thing comes after that is overview of a cell. We basically took two different kinds of plant cell and animal cell, one from each. In the typical plant cell we took onion cell, and typical animal cell we took as human cheek cell. Okay. And we observed the differences, that a large vacuole is present in plant cell and absent in animal cell. Similarly, cell wall is absent in animal cell and present in plant cell. Centriole is absent in plant cell and present in animal cell. And the fourth one is plastids or chloroplast is present only in plant cells and absent in animal cells. Now both plant cell and animal cell together are called as eukaryotic cells. Eu means true and karyon means nucleus. So it means true nucleus. They have a membrane bound, they have a nuclear envelope covering the nucleus. Okay. Now next we talked about bacterial cell, okay, or basically prokaryotic cell. We discussed the features of the bacterial cell. Okay. First thing, we will start from the outermost structure of bacterial cell, which is glycocalyx. Glycocalyx basically further has two layers, one which is solid glycocalyx or called capsule, then loose glycocalyx or also called slime layer. Capsule's work is protection from WBC, and it also helps in colony formation. Then slime layer helps the bacteria to stick to certain things, and it also helps to prevent bacteria from drying out. Okay, next cell wall of bacterial cell is composed of peptidoglycan. Then there is also periplasmic space present between cell membrane and cell wall, which acts just like lysosomes of eukaryotic cell. It has digestive enzymes present in it, which helps in digestion. Next comes mesosomes. Mesosomes are basically invagination of cell membrane in the bacterial cell. Okay, its main function is respiration, but it also helps in DNA replication, cell division, DNA copying, cell wall material synthesis, protein and enzyme synthesis, etc. Okay, mesosomes has been compared with the mitochondria of the animal or can say eukaryotic cell, because mitochondria also helps in respiration. Okay. Next we studied about the longer pili and shorter pili. Longer pili and shorter pili are made up of pillin protein. Longer pili is also known as sex pili, okay, and it helps in conjugation. And shorter pili is also known as somatic pili, which helps in attachment. Okay. Next is flagella. In bacterial cells, it is made up of flagellin protein, which is contractile in nature and it helps in locomotion. Okay. Next is DNA. We studied also about their DNA. Bacterial cell DNA is made up of bacterial cell DNA is basically double stranded circular DNA. Okay, and it also has an extra DNA called plasmid. It has resistance from specific antibiotics and fertility things also. Okay. One more important thing, that bacterial cells have 70S ribosomes, okay, whereas animal cells have 70S and 80S, both types of ribosomes. Then we studied the differences between prokaryotes and eukaryotes. First difference is nucleus. Nucleus is basically double stranded circular DNA in prokaryotes and double stranded linear DNA in eukaryotes. Then second thing comes is compartmentalization. Compartmentalization is basically absent in prokaryotes because they lack membrane bound organelles, whereas it is present in eukaryotes because they have membrane bound organelles. For example, mitochondria and chloroplast are double membrane bound organelles and nucleus is also double membrane bound. Now lysosomes, ER, endoplasmic reticulums, Golgi apparatus, etc. are single membrane bound, and ribosomes and centrosomes and nucleolus, they are membraneless. Now third thing comes is vacuole. Okay. Now sap vacuole is absent in prokaryotes and sap vacuole is present in eukaryotes. Okay. Whereas gas vacuole is present in prokaryotes. Okay. Now next difference is in chromosomes. They are basically double stranded circular DNA in eukaryotes and double stranded linear DNA in prokaryotes. Nucleus, in nucleus prokaryotes lack a nuclear membrane, whereas eukaryotes have a nuclear membrane. Okay. Digestive enzymes. Okay, next difference is digestive enzymes. Prokaryotes basically have mesosomes and cytoplasm, and eukaryotes have mitochondrial and cytoplasm. Now next thing is cyclosis. Cyclosis is basically, next thing comes is cyclosis. Cyclosis is basically circular movement of cytoplasm, which helps in transportation of materials inside the cell and also helps in mixing of materials. Okay. Cyclosis is absent in prokaryotes and present in eukaryotes. Okay. Last difference comes is ploidy. Ploidy is basically the number of set of chromosomes present inside the cell. Okay. So prokaryotes have basically one set, or I can say haploid, and eukaryotes are haploid, diploid and as well as polyploid. Now examples of prokaryotes are cyanobacteria and mycoplasma. Now examples of eukaryotes are fungi, protista, animals, etc. Next we studied about Gram staining process, that with bacteria which take up the color are known as Gram positive and which do not are known as Gram negative. Then we discussed about the sizes of different types of cells, like RBC is of 7 nanometer, eukaryotic cells are 10 to 20 nanometers, bacterial cell are 3 to 5 nanometers, and typical bacterial cell is 1 to 2 nanometers also. Now mycoplasma is 0.3 nanometer, then PPLO, pleuropneumonia like organisms, are of 0.1 micrometer, sorry, not nanometer anyway, it is micrometer. Then largest isolated cell is ostrich egg cell, then longest cell is nerve cell, and also that their shapes are according to their functions they perform. Okay, like RBC are round and biconcave, WBC are in the structure of amoeboid, then tracheids are elongated, columnar epithelial cells are long and narrow, nerve cells are long and branched, okay, etc. like this only. Then we discussed about the cell membrane. Cell membrane is also known as unit membrane, biomembrane, plasma membrane, or plasma lemma. Okay. Now, after 1950s, there were information about cell membrane coming. It was because of discovery of electron microscope after the 1950s. Okay. So the first model we got was sandwich model, but it was rejected because it does not tell us about the permeability of the cell membrane. Okay, cell membrane is basically thin, flexible, universal, and semi-permeable or selectively permeable in nature. Now we will discuss about its chemical composition. Lipids are present in the 40%, proteins are 52%, and carbohydrates 8 to 9% in the RBCs. Okay, but in other cells, proteins are present are 58%, lipids are 40%, and carbohydrates 1 to 2%. Next model was fluid mosaic model, which was given by Nicholson and Singer scientists. This is the model which is now accepted in the whole world mostly. Okay, we studied about this model also. It is also called as Gulab Jamun model or icebergs of protein floating in the sea of lipids. We studied about its structure, we studied about the proteins and also its functions like channel protein or carrier protein and tunnel protein, structural protein, etc. Channel or tunnel protein helps in passive transportation and carrier protein helps in passive as well as active transportation. Structural protein helps to provide structure to the cell membrane. Then what are the types of transports? Basically there can be of two types, active transport and passive transport. Okay, in passive transport there is no requirement of energy and movement of particles is from higher gradient to lower gradient, and active transportation is where energy is required and transfer of molecules or substances are against the gradient concentration. It is basically from low gradient to high gradient. Okay, now passive transportation can further be classified into two types, simple diffusion and facilitated diffusion. In simple diffusion, diffusion of materials takes place across the cell membrane easily. For example, gaseous exchange, and specially molecules or substances must be very small, and another thing that they should be non-polar or neutral. They should not contain any charge. And in facilitated diffusion, there is a requirement of facilitator protein. Okay. Now there are two types of active transportation also. Basically it happens in bulk transportation, phagocytosis and pinocytosis. It comes under endocytosis, and another type is exocytosis. In phagocytosis, cells take up solid materials, okay, or basically there is intake of solid materials in large quantities, so it is known as phagocytosis. And when cell take up liquid substances in large quantities, it is known as pinocytosis. Okay. Now next comes is exocytosis or amniocytosis, or this is also known as cell vomiting. In this process, cell basically excrete out waste products or can say unwanted things out of their body. Okay. Now comes what is the significance of fluidity in the cell membrane. It is basically helps in growth, helps in cell division, helps in endocytosis, exocytosis, etc. Then also it helps in interdigitations, which helps us to provide flexibility, and also secretion. Okay. Now we also discussed about the cell wall. Cell wall in plants are basically composed of cellulose, hemicellulose, pectins, and proteins. Cell wall of fungi is composed of chitin. Cell wall in algae is basically composed of cellulose, mannans, galactans, and other minerals such as calcium carbonate. Okay. Now cell wall of bacteria is composed of peptidoglycan, as I have already discussed. And now what are the functions of cell wall in basically plant cell? Okay. It helps in cell to cell interaction. It provides a mechanical barrier. It acts as a barrier for undesirable macromolecules. It also protects the plants from infection and mechanical damage. Okay. And it also provides a shape and size to the cell membrane. Okay. That's all for today's. Thank you.